Welcome everyone to the WebEVTalk. So today we have speakers and also hosts from Japan, Professor Takahiro Ochiya and also Fumihiko Urabe from Japan. So please, Ochiya Sensei, please go ahead and introduce Fumihiko.
Okay. All right. Thank you very much, Caroline. So hello everyone. My name is Takahiro from Tokyo. And today I'm very happy to introduce Dr. Fumihiko Urabe from Tokyo, Japan. Fumihiko is graduated from the Jikei University School of Medicine, Tokyo in 2012, and then completed his residency at the same institution. He joined my team at National Cancer Center Research Institute in 2016 and obtained his PhD at 2020. Yeah, Fumihiko, one of the best PhD students in my lab, he's so excellent. He's currently appointed as clinical associate of the Jikei University School of Medicine and works as a urologist. His research focuses on miRNA regular biopsy and, of course, extracellular vesicles in prostate cancers. Today, Fumihiko is going to talk the potential of clinical applications of EVs and miRNAs in urological cancer management. Okay, Fumihiko, please.
Thank you for introduction, and good evening and good morning, everyone. Thank you for giving me this great opportunity to present my project. Today, I would like to show the results about extracellular vesicles and miRNAs in urological cancer. This is my COI, and as introduced by Professor Ochiya Sensei, this is my profile. My name is Fumihiko Urabe, a Japanese urologist. I graduated from the Jikei University School of Medicine in 2012 in Japan. After graduating from the Jikei University, I work as an urological resident in my hospital. And I joined Ochiya Sensei's lab in 2016. During my PhD training, my research topics are extracellular vesicles and miRNAs in urological cancers. Fortunately, I could receive PhD last September, and now I work as a staff urologist in University Hospital. In Japan, COVID-19 is still going around, so it is hard time for doctors to see patients. But in this difficult time, thanks to WebEVTalk, I could get a great opportunity to share my research work from my country, and especially in front of my laptop. Indeed, and until 30 minutes before, I work at outpatient department in my university hospital. Thanks to web meeting, I could attend the meeting soon after my duty work. And in my PhD training, I belong to National Cancer Center, Tokyo, Japan. And my professor was Ochiya Sensei. As you know, he's a famous EV researcher in Japan. And this is a group photo in 2018, and this is me. Then let's start my presentation. As you know, extracellular vesicles, EVs, are small bilayered vesicles and secreted from almost all types of cells. EVs have long been thought of as no more than a garbage can for cells to discard unwanted components. However, in 2007, Professor Jan Rottbauer reported EVs contain miRNAs and messenger RNAs and suggested their function in recipient cells. From then, many researchers started focusing on EVs. Until now, EVs are reported to be contain pathogenic components such as proteins, miRNAs and messenger RNAs and DNA fragments, and through transfer, these molecules via EVs, and the recipient cells receive the EVs, and the EVs contributed to intercellular communication. In Japan, Dr. Kosaka, one of my mentors, reported the function of miRNAs in EV's recipient cell in 2010 when he had belonged to Ochiya Sensei's lab. And this is one of the first reports to show the function of EVs. And because then I'm a clinician, urologist, I wanted to try clinical useful research topics. So I decided my research goal is to establish novel EV-based therapeutics for urological cancers. Therefore, I selected these 3 topics as my research. From EV research, establishing novel biomarkers, clarifying the molecular mechanism of bone metastasis, and establishing EV therapeutics by targeting EV biogenesis. From the next slides, I would like to introduce these topics. In this study, we performed an experiment according to ICEP position paper, MICEP 2018. And the first one is establishing novel biomarkers to diagnose urological cancers. As you know, miRNAs are short non-coding RNAs consisting of 19 to 25 nucleotides. miRNAs regulate gene expression by inhibiting the translation of target messenger RNAs. Now, total of 2,588 miRNAs are identified. In 2005, a Nature paper reported miRNA expression of tissue can classify human cancers. This finding highlights the potential of miRNA profiling in cancer diagnosis. Recently, cycling miRNA have gained attention. The important point is that miRNAs can be released from various types of cells, including cancer cells, and can be stably present in the circulation, and can be taken up by other cells to assist cancer progression. Therefore, cycling miRNA profile is a good candidate of less invasive biomarker for cancer diagnosis. About half of cycling miRNA is reported to be protected by EVs. There's tough relationship between cycling miRNA and EVs. And so in 2014, Ochiya Sensei's lab group launched the largest scale national project. The aim of this big project is to develop the novel diagnostic technology for certain types of solid cancers and dementia using cycling miRNA profiles. In these 13 types of solid cancers, as urological cancers, prostate cancer, and bladder cancer were contained. To be exact, this project is not for pure exosomal miRNA study, but the most of about half of miRNAs are protected by EVs. And to show the potential of EV biomarker, I introduced this study in the first part of my presentation. From the next slide, I will briefly show the results. The first one is prostate cancer. Prostate cancer is the most frequently diagnosed cancer in men and the third leading cause of cancer-related deaths in men in the United States. And its incidence and the mortality are also increasing in Japan. Digital rectal examination and PSA monitoring are the standard methods of prostate cancer screening. However, the accuracy of these methods for detection of prostate cancer is limited. DRE is subjective test and PSA has low specificity. Therefore, this method may lead unnecessary prostate biopsy. Indeed, in the United States, PSA testing is estimated to lead to approximately 700,000 unnecessary biopsies for prostate cancer every year. So, in daily clinical practice, the development of efficient and less invasive biomarker is required. So in the present study, we aim to establish a diagnostic model composed of cycling miRNAs to detect prostate cancer in men with suspected prostate cancer. This is the overview of this study. Serum micron expression profiles of all samples were comprehensively analyzed using miRNA microarray. Samples were randomly assigned to discovery, training, and validation set. In each set, in discovery set, we selected candidate prostate cancer specific miRNAs. In training set, we constructed the model. In validation set, we validated its performance. Participant characteristics are shown here. All clinical factors are well balanced between discovery, training, and validation set. For the miRNA microarray analysis, total RNAs are extracted from 300 micromilliliters of serum samples. All miRNA protocols are perfectly standardized and strictly controlled in our project. In discovery set, the expression level of 2,588 microns were comprehensively analyzed. And finally, we identified 60 microns that were specifically upregulated in prostate cancer samples and 2 microns that were specifically downregulated in prostate cancer samples. In the training set, the best combination of identified 18 microns were exploring using Fisher linear discriminant analysis with leave one out cross-validation. The best discriminants for each number of microns were listed, and finally, the model showing the best AUC with the least number of microns was selected. The solution of the index more than or equal to 0 indicated presence of prostate cancer, and whereas an index less than 0 indicated the absence of prostate cancer. Finally, the combination of miR-17 and miR-1185 was selected as the best discriminant model. In validation set, we could confirm the performance of our diagnostic model composed of 2 microns with high sensitivity and specificity. In addition, as shown in the figures, the model shows high performance for each prostate cancer grade. These data show our model is useful for diagnosis of any stage of prostate cancer. In another topic, we focus on bladder cancer. Lack of sensitivity and specificity, or specificity of urinary test, many suspected patients performed cystoscopy. But cystoscopy is painful and may cause urinary infection. So, the development of efficient and less invasive biomarker is urgent in bladder cancer screening. As same as in prostate cancer, we perform comprehensively analyze the serum micron expression profiles of about 1,000 samples using miRNA microarray. In bladder cancer, we divided these samples into 2 groups, training set and validation set. In training set, we selected bladder cancer specific miRNAs and constructed the best discriminant model. And in validation set, we validated its performance. In bladder cancer model, we could establish a high sensitivity and specificity model, a combination of the 7 microns. This is a training set, and this is a validation set. And this is a conclusion. And the present study is a large scale performed to date, and all samples were comprehensively analyzed using miRNA microarray. The result indicated that evaluation of cyclin microRNAs is a feasible method for detecting prostate cancer and bladder cancer. These models could reduce the number of unnecessary biopsies with cystoscopy and improve accuracy of cancer diagnosis. Now we are performing the prospective studies to confirm the diagnostic performance of these models. And the second topic is revealing a novel molecular mechanism of progression of metastasis. Today's EVs are reported to transfer functional molecules into recipient cell and contribute to intercellular communication. Especially in the field of cancer research, EVs are reported to impact the multi-step process of cancer progression. For instance, cancer-derived EVs induce angiogenesis and modulate the immune system and provide a drug resistance. Today, the number of members of Ochiya Sensei's lab have reported several interesting studies of EV roles in cancer metastasis in major journals. Dr. Kosaka reported EVs from breast cancer promote angiogenesis and contribute to lung metastasis. Dr. Ono reported EVs from MSC induce dormancy of breast cancer and contribute to long-term recurrence. Dr. Tominaga and Dr. Yokoi reported EVs destroy blood-brain barrier or peritoneum and promote cancer progression. Dr. Tominaga focused on breast cancer and breast cancer brain metastasis, and Dr. Yokoi focused on ovarian cancer. When I think the famous metastatic side of urological cancer, bone metastasis and prostate cancer first come into mind. So I decided to focus on prostate cancer and bone metastasis. Firstly, I show the background of this project. Prostate cancer is the most frequently diagnosed male cancer in Western countries. In clinical, bone is the most metastatic site in advanced stage prostate cancer, and it is not only curable, but also devastates patients' quality of life. Therefore, bone metastasis is a big clinical topic. This is a schematic of bone metabolism. Osteoblasts and osteoclasts interact with each other, and continuously, bone is always remodeling. However, once prostate cancer metastasizes to bone, prostate cancer educates osteoblasts and osteoclasts to benefit the growth in the metastatic site. This concept, vicious cycle, is widely accepted, but underlying mechanism of bone metastasis remains obscure. Indeed, in clinical, LAMP2 inhibitor is a famous option to treat advanced stage prostate cancer patients, but we can't completely inhibit progression on bone metastasis. This suggests an unknown molecular mechanism will be present in the mechanism of bone metastasis. Therefore, we hypothesize that the prostate cancer cell-derived EVs contribute to progression of bone metastasis via promoting osteoclast differentiation. Firstly, we checked whether prostate cancer-derived EVs promote osteoclast differentiation. Cells positive for trypsin with 3 or more nuclei were considered osteoclasts, and the number of osteoclasts were counted. When we add only EVs to osteoblast precursors, we could not find any osteoclasts. However, when we add EVs in the presence of LAMP2, there is a synergistic effect compared to adding only LAMP2. This is only LAMP2 and their combination of LAMP2 with EVs. To evaluate the effect of EVs derived from normal prostate epithelial cells, we collected EVs from normal prostate epithelial cells and performed the same experiment. As shown in the orange bars, we found that normal prostate epithelial cell derived EVs did not promote osteoclast differentiation, even in the presence of LAMP2. So, we found the prostate cancer cell derived EVs specifically promote osteoclast differentiation. Next, to find a specific protein on EVs to promote osteoclast differentiation, we performed this experiment. We compared prostate cancer cell-derived EVs and the normal prostate epithelial-derived EVs by proteome analysis. We selected proteins whose expression level is higher in prostate cancer-derived EVs. Candidate proteins were picked up as shown in the light table, and sRNA of these genes were transfected to prostate cancer cells. And from the sRNA transfected prostate cancer cells, we collected EVs. The amount of proteins in EVs was adjusted, and EVs were added to osteoporosis precursors baseline group. After 96 hours, we checked the differentiation by trap stain. We performed 3 times screening, and finally, we found one molecule, CDCP1, contribute osteoporosis differentiation. This is immunoelectron microscope, and we confirmed that this molecule is located on EVs. The black dot is an CDCP1. This is an EVs from prostate cancer. This is EVs from a normal prostate epithelial cell. To further test with the CDCP1 promote osteoclast differentiation crystallogenesis, we stably overexpress the CDCP1 in HEC 293 cells with low basal level of CDCP1. CDCP1 detected in the EVs isolated from the culture medium of CDCP1 overexpressing HEC 293 cell, and it was confirmed by immunoblotting and immunomicroscope. CDCP1 overexpressing and HEC 293 derived EVs accelerates osteoclast formation compared to control. These findings indicated that CDCP1 overexpressing HEC 293 derived EVs promote osteoclast formation differentiation. Next, we evaluated expression of this molecule in clinical sample. The expression level of CDCP1 in plasma-derived EVs from prostate cancer patients were examined. The blue ones are localized prostate cancer patient, and the red ones are bone metastatic prostate cancer patient. CDCP1 was detected in plasma-derived EVs from advanced stage prostate cancer patient. Especially, this sample is from patients with multiple bone metastases. Therefore, we think that this molecule on EVs could be a biomarker to predict the bone metastasis. And this is summary of the second topic. In this study, we found that the prostate cancer cell-derived EVs promote osteoclast differentiation. And we found that CDCP1 is important to provide these phenotypical changes. CDCP1 was detected in EVs from advanced stage prostate cancer patient. Therefore, we believe this molecule could be a novel therapeutic target or biomarker. The last topic is revealing novel molecular mechanism of biogenesis of EVs. As we have referred, EVs are reported to impact the multi-step process of cancer progression. Therefore, to inhibit intercellular communication via EVs in tumor microenvironment, it can be a novel therapeutic strategy. Several ideas of inhibiting intercellular communication via EVs are proposed. Inhibition of EV secretion, elimination of targeting EVs, and disruption of EV absorption. In this study, I focus on inhibition of EV secretion. In a previous work, Dr. Nishida reported therapeutic strategy targeting cancer-derived EVs. In this study, she targeted CD9 and CD63 located on EVs to exclude EVs from xenograft model. Treatment with specific human-specific anti-CD9 and anti-CD63 antibodies significantly decrease the metastasis to lung, lymph node, lung, and thoracic cavity, although no obvious effects on primary xenograft tumor growth were observed. This study suggests that the potential of the removal of EVs as a novel strategy for cancer therapy. And another study, we found that neutral sphingomyelin 2 regulates EV biogenesis and attenuation of this gene inhibit EV circulation and suppress angiogenesis and metastasis in breast cancer xenograft. However, the downregulation of neutral sphingomyelin 2 did not inhibit the circulation of EVs from prostate cancer cells. Therefore, to establish EV targeted cancer-specific or disease-specific therapies, the identification of disease-specific mechanisms of EV circulation is required. Especially, we found that prostate cancer cells circulate more EVs than normal prostate epithelial cells using the ultracentrifugation and nanoparticle tracking analysis. However, the precise mechanism of EV circulation in prostate cancer remains obscure. These are the first questions to start this experiment. We don't clearly know what kind of genes regulate biogenesis of EVs in prostate cancer. Therefore, to review the molecular mechanism of EV biogenesis in prostate cancer, it will contribute to develop a novel therapeutic strategy for prostate cancer. The gold standard method of EV collection is their ultracentrifugation. However, it takes much time to isolate EV fraction, and thus screening the genes regulating EV biogenesis was difficult. In 2014, we developed the novel EV detection method. This is our original assay called ExoScreen. ExoScreen is based on amplified luminescent proximity homogeneous assay using photosensitizer beads. In this system, we employed two types of antibodies with donor beads and acceptor beads. The donor beads excited with laser at 680 nanometer, resulting in the release of singlet oxygen. The singlet oxygen stimulate the amplified fluorescent signals in the acceptor beads, when the beads are within 200 nanometers of the captured EVs. As shown in the below figures, we can evaluate the amount of EVs by using this combination of antibodies. Using a CD9 antibodies, using CD9 CD63 antibodies, using a CD60 antibodies. Using this ExoScreen, we could easily evaluate the amount of EVs and perform the high throughput screening to detect the mechanism of EV biogenesis in prostate cancer. This figure shows the correlation between signal intensity measured by ExoScreen and amount of conditioned medium. We could evaluate the amount of EVs without purification step and reveal that ExoScreen is sensitive enough to detect EVs from only 1 nanometer of cell culture medium. In addition, in this study, we hypothesized that miRNAs regulate EV circulation and used miRNA library to screen the genes regulating EV biogenesis. First, we confirmed that CD9 and CD63 are contained on EVs from prostate cancer cells by immunoblotting. In this study, we mainly used the PC3M, which is one of the major prostate cancer cell line, and correlation of ExoScreen measurements using CD9 antibodies, CD9 CD63 antibodies, CD60 antibodies with a particular number of EVs in dilution series. These results indicated that we can use CD9, CD63, and CD60 antibodies to evaluate the amount of EVs without purification. To find a specific miRNAs which regulate the EV circulation, we established and performed high-throughput screening. We transfected each miRNA to prostate cancer cell, and using a miRNA library. And from the transfected cells, we evaluated the amount of EVs using ExoScreen assay. And to exclude the effect of miRNAs on cellular population, we performed cell growth assay, MTT assay. EV circulation was calculated through the ExoScreen and cell growth assay, and the value were evaluated as a fold change relative to negative control and positive control. In this study, we used positive control, TSG 101. The goal of this study is to find a prostate cancer specific gene to regulate EV biogenesis. In this period, we selected miRNAs which reduced amount of EVs after transfection. In the first screening, we used the CD9 antibodies. We performed a 3-time screening, and finally, we identified 30 miRNAs, and then using a CD9 antibody. Then, to further validate the initial screening, the circulation of CD60 positive EVs and CD9 double positive EVs were assessed by ExoScreen. And finally, in this screening, we found miR26A and miR194 candidate miRNAs which regulate EV circulation. From public database, we selected miR26A because the expression level of miR26A is lower in prostate cancer than normal prostate epithelial cell tissue. Therefore, in this study, we focus on miR26A. In addition, using nanoparticle tracking analysis, we could validate the particle number of EVs secreted by each prostate cancer cells, also decreased by transfection of a miR26A mimic in PC3M and PC3 cells. Therefore, we could validate miR26A regulate EV circulation in prostate cancer cells. Then, to further elucidate the molecular mechanism of miR26A in EV circulation, we screened the target genes of miR26A. Messenger RNA microRNA analysis in PC3M and PC3 was performed after transfection of miR26A mimic or control. In combination with the result of target scan, 88 candidate genes were identified by this assay. Then, to select genes regulating EV circulation, we performed high-throughput screening again using siRNA of these candidate genes, ExoScreen, and cell growth assay. In this screening, we found 4 genes, SHC4, PFDN4, CHRDC1, and PRKCD were identified as candidate genes regulating EV circulation in prostate cancer. Using nanoparticle tracking analysis, we could validate the particle number of EVs secreted by each prostate cancer cells, also decreased by the downregulation of these 3 genes, SHC4, PFDN4, CHRDC1. Then, using a public database, the expression level of PFDN4 and CHRDC1 was significantly upregulated in prostate cancer tissue compared with normal tissue. Finally, we examined the downregulation of EV secretion can inhibit prostate cancer cell progression in vivo. Initially, stable SHC4, PFDN4, and CHRDC1 depletion was established by using each short hairpin RNA. And we validated these stable lines secrete less EVs than control PC3M cells. Each stable line were subcutaneously injected into mice, and 3 micrograms of EVs from control prostate control PC3M cells was injected into tumorally every other day for up to 28 days. Then tumor size and the tumor weight were measured. These are the results of in vivo study. The tumor xenograft of noodle mice injected with prostate cancer EVs partially rescued the tumor size and tumor weight of genes and depleted prostate cancer xenograft. The red ones are rescued group. And this is summary of this study. In this study, we could find a normal miRNA regulating EV secretion in prostate cancer. And SHC4, PFDN4, CHRDC1 are expected to be beneficial for the inhibition of cell-cell communication via EVs in prostate cancer. As I have shown during my PhD training, I focus on these topics. Some projects are still undergoing. However, I could reveal several interesting findings in urological cancers. And this is summary and the future strategy. As I described in my review paper, further research and development are required for clinical application, but the clinical utility of EVs is promising, and their endless possibility in the field of EV research, and especially, I think especially in urological cancers. I enthusiastically hope that the clinical use of EVs will contribute to urological cancer management and treatment in the near future. The slides are list my collaborators, and thank you very much for your kind attention.
Okay, thank you, Fumihiko. Very nice talk. So now, would you please accept a couple of questions from the audience? Okay. All right, so please write down your question. Okay, so one question from the audience. Okay. Boya-san, thank you very much for the, okay. Urabe-san, can you follow to read? Have you ever used saliva, urine, or other type of biofluids for the regular biopsy to analyze microRNA? The question.
In my study, I mainly focus on serum. So I haven't checked whether these microRNAs can useful for urological cancer detection. But I think, especially in urine, it's a very effective biofluid, because especially in bladder cancer based in urine for several hours before out. So I think urine could be a great marker. Recent reports and many reports and reveal that urine is a very effective biomarker, could be biomarker.
Okay, so please ask questions. If you mute off and video on, so you can speak. Okay. Someone else?
I think I have one. Hi, Fumihiko, a great talk. Thank you so much. I'm just wondering whether you have in mind a strategy on how to target prostate cancer specifically. I mean, you have a few candidates to target, but do you have sort of how you want to target them?
You mean your question is that my third part?
Mm-hmm. The therapeutics and the therapeutic strategy, I think.
Mm-hmm. Okay, thank you for your question. In fact, in this study, we could not find a exact molecular mechanism of these genes, but SHC4 is related to the LAB11, which is a famous gene to promote EV circulation, and CHRD7 is related is a co-chaperone of histone protein 90. It's also related to EV circulation. So these genes have we could find these genes could regulate the EV circulation, but in this study we could only find that these genes are regulated in prostate cancer, but we think that these genes can be a therapeutic target. But as the next step, and we want to elucidate the exact molecular mechanism of these genes, and if possible, I want to make drugs to target genes to treat prostate cancer. And especially this prostate cancer cell line is an advanced stage prostate cancer, because the PC3M is get from the almost CRPC, castration-resistant prostate cancer. So I want to elucidate the mechanism of these genes, molecular mechanism of these genes, and could use these genes to treat an advanced stage prostate cancer patient.
Actually, yeah, that's really interesting about that osteoclast differentiation. So have you also looked whether it's EV specific, or if there's actually a possibility that cell-cell interaction, like direct interaction, can also induce osteoclast differentiation?
Your question is that only CDCP1 on EVs can promote osteoclast differentiation? Or,
I mean, is it really specifically through EVs, or is it possible that the prostate cancer, if they're in the close proximity with the osteoclast precursor cells, they can also induce this differentiation?
In this study, we could check and we collect the EVs from prostate cancer cell, and the CDCP1 is upregulated in prostate cancer cell-derived EVs, and it could promote osteoclast differentiation in the presence of LANCRO, and upregulated HEC293, upregulation CDCP1 on HEC293 cell, and derived from this cell can also promote osteoclast differentiation. So I think that we could then confirm that the CDCP1 on EVs can osteoclast differentiation, but I don't check yet whether the cell-cell communication is also on prostate cancer cell. The CDCP1 on prostate cancer cell can also promote osteoclast differentiation. We couldn't examine yet.
Okay, thank you. So another question, Alicia. So thank you very much for your comment. So Fumihiko, can you read Alicia's comment? She also found the CDCP1 in the PC3 cells, EVs, and compared to the normal prostate cells. So Alicia, if you're okay, so would you mute off and discuss with Fumihiko on this matter? Hello, Alicia.
Hi.
Thank you.
Yes, I found this protein some years ago in a proteomic screening that we did in the lab. And then I compared it to another normal prostate cancer cell line, the RWPE. And I also found that this protein was, the levels of expression were higher in this in the PC3 cell line. So I think this protein was quite interesting because it's also a membrane protein, so you can detect it on the surface. And also it's a protein that has been related to other types of cancer. So I think it was an interesting protein. But when I did it studies in urine, comparing healthy versus prostate cancer, I couldn't find this protein overexpressed in the urine of prostate cancer vesicles. Okay. Vesicles from prostate cancer. So, but at least from I think this protein is quite interesting.
That's great. Thank you very much, Alicia. So, Fumihiko, what do you say?
In this and our study, in our study, we checked the expression level of CDCP1 from the advanced stage prostate cancer patient. So I think that it has a big tumor in their bodies. So much secreted EVs are circulating in their body. So we can easily find CDCP1 by plasma. But I think that very early stage of prostate cancer to detect them by CDCP1 might be difficult, I think. I haven't checked the urinary expression.
Okay, thank you very much.
Okay, so Alicia, so what do you think? We are now working on the molecular mechanisms of CDCP1 EVs. If they transfer to the recipient cells, such as osteoclast cells, maybe transfer some signals to the osteoclast cells. But still, we don't know exact molecular mechanism. Do you have any idea, or do you have any data on the CDCP1 related target molecular mechanisms, right?
No, I haven't worked now with this protein for a while, since I was more interested in the biomarker use of the protein. So I haven't investigated possible mechanisms of this protein in the progression of prostate cancer.
All right. Thank you very much for your valuable comment.
Welcome.
Thank you.
So other questions? Okay. Maybe not. Maybe so. Carolina-san, please.
Okay. Yeah, thank you so much again, Fumihiko and Ochiya Sensei. It's wonderful sessions today and a lot of update from Japan.